Okay, so as promised, finally, here is the short EdPuzzle video on gene regulation in prokaryotes. So in class, we were talking about how eukaryotes, that includes us, regulate which genes are expressed and when through the use of activator and repressor molecules to regulate how often or whether it is available at all, the promoter for a particular gene is available to be transcribed, and the activity of RNA polymerase, basically how often RNA polymerase is attracted to a promoter, which is going to affect how often it transcribes a particular gene. The same basic principles are in effect here for prokaryotes, but they organize their genes differently, and so that necessitates some different mechanisms to control which genes are expressed and when. Okay, so prokaryotes, we know, organize their genome a little bit differently than eukaryotes, right? So in prokaryotes, the genes that code for the enzymes that are all involved in the same metabolic pathway are grouped together on the chromosome. And by together, we mean like right next to each other. And they actually share a promoter so that when RNA polymerase transcribes, and binds to that particular promoter, it doesn't just transcribe one gene, it transcribes all the genes that are next to that promoter. So that means that prokaryotes are controlling transcription of all of these genes at the same time. So they're either, all transcribed or none of them are transcribed. It's really kind of an all or nothing kind of situation. And so in prokaryotes, they control transcription of all of these genes that are attached to the single promoter by the use of a DNA sequence called the operator. And in some ways, this is similar to when we were talking about in class, we talked about eukaryotes and we talked about control elements that are grouped together into those enhancer regions. This is the same kind of idea. So control elements are DNA sequences that bind to activators or repressors in eukaryotes. And obviously activators, increase transcription and repressors decrease or turn off transcription because of their effect on the promoter region. Prokaryotes, same kind of idea. Rather than having maybe multiple control elements, they have a single DNA sequence called the operator. And if the operator is bound to something, it blocks transcription and the gene is turned off. And if the operator is free, basically not bound to anything, then RNA polymerase can access the promoter and the gene is turned on and transcription can happen. So basically what we have the operator sequence, we have the promoter, and then we have all the genes that are next to the promoter that are all involved in the same metabolic pathway. And we group those together and refer to them, to all those three things, as an operon. And so we may talk about, for example, the lac operon. This is an operon that contains the genes that allow bacteria to break down lactose. And so it controls all of the enzymes that are involved in the process of breaking down lactose, which is a multi-stage process, right? Lactose being a more complex sugar than glucose. But you have multiple enzymes that are involved in that pathway. And if you have lactose present, you want to make those enzymes so that you can break down lactose and get the energy out of it, just like you would break down glucose or any other carbohydrate. So you're going to transcribe the genes. They're all next to the same promoter. And then there is one operator that controls transcription for all of those genes together. Okay, so As I mentioned, we're regulating transcription based on whether the operator is bound to something or whether the operator is free, meaning it's not bound to something. And the something is going to be something that's called a repressor. And if the operator is bound to the repressor, then transcription cannot happen. And again, if the operator is free, then transcription of all of the genes involved in the operon is going to proceed. So as you can see on the little visual here, right, the repressor is, if I can find my little doohickey here, my little pen, okay. So the repressor molecule is produced by a separate gene with its own promoter. So here's the repressor gene, here is the lac I, right, labeled here. So that's the regulatory gene. The regulatory gene codes for the repressor molecule. And you'll notice that the regulatory gene has its own promoter here in green. I'm going to sort of underline that right there to the left of the lac I gene. And there's no operator. So this basically means that there is no way in bacteria that they can block transcription of the regulatory gene. So it's always on. Now you also can't really turn it up too much. So it's always on at a low level, meaning that this particular gene is always being transcribed. And as we can see, right, we've got the messenger RNA, and we know, right, that's the 5' to 3', right? So here's the start part of that one. That's going to go to the ribosome, and then the ribosome is going to make the repressor molecule. And the repressor molecule is in its active form is going to bind to the operator, and it's always the active form that's bound to the operator. And if the operator is bound, then as you can see on this visual, right, RNA polymerase is trying to access the promoter, but it can't, right? It's being blocked. So you have basically no transcription is happening, right? So no RNA gets made in this particular situation. It's always the active form of the repressor that's bound to the operator. So those regulatory genes are always on. They're always making the repressor molecule at a low rate. The repressors, you notice I said the active form of the repressor, and you're probably like, well, is there an inactive form? And the answer is yes. So repressors are allosterically regulated molecules. And as you guys remember from way back when, right, exam 2, we were talking about enzyme regulation. Allosterically regulated enzymes have an active form and an inactive form. And so what can happen is either they require something to make them active, and if the something is missing, then they are in the inactive form, or they exist in an active form and an inactive form. They spontaneously go back and forth between, and you have activators that lock them in the active form and repressors that lock them in the inactive form. So with operons, we're looking at the first example. So you have the repressor is either produced in the active form, and it requires another molecule binding to it to essentially turn it off in a kind of non-competitive inhibitory fashion, or your operon is produced in the inactive form, and it requires being bound to another molecule in order to turn it into its active form. But it can exist in both forms, and then of course that's how they regulate, they being bacteria, regulate transcription. If you have an active form of the repressor, then it binds to the operator and blocks transcription. If the repressor is in the inactive form, it cannot bind to the operator, and therefore we have transcription happening. Okay, so it is, as we mentioned, it's always the active form of the repressor that is going to bind to the operator and turn the gene off. And so the fact that it's an active form of the repressor that binds to the operator, we refer to that as negative gene control. So you can think about negative gene control as sort of like an on-off switch, right? So the operator basically, the repressor basically acts like an off switch. In its active form, it binds to the operator. If the operator is bound, transcription can't happen, and your whole operon is turned off, right? You can't transcribe any of the genes in that metabolic pathway. So based on that, we have two different kinds of operons. We have an inducible operon. And so I always think about inducible, right, as you're inducing labor, you're starting labor. So with drugs. So this is something that has to be induced. It has to be turned on. So an inducible operon is actually usually off. So the operon is normally turned off, meaning that the repressor, right, is normally produced in the active form, right? Because it's always the active form of the repressor that that binds to the operator, right? That's always true. Whether inducible operon, repressible operon, it's always the active form of the repressor that binds to the operator, right? If the repressor is not active, then it doesn't have the right shape to bind to the repressor, to bind to the operon operator. I'll get it right eventually. It doesn't have the right shape to bind to the operator, and therefore the operon is on, right? If the operator is free. So in an inducible operon, it's normally on, which means, right, that our repressor molecule here is produced, and this is kind of like why it looks sort of shiny. It sort of looks like it's glowing. So it's produced in the active form. So that means that it's going to be, without any alteration or having anything else bind to it, it's going to bind to the operator, and transcription is going to be blocked, meaning the operon is off, right? And so we have to, these kinds of operons, we have to induce the change. We have to essentially turn the operon on, and in inducing the operon, we have to deactivate the repressor, right? So the repressor needs to be bound to something else, which is a molecule that we call the inducer. It's inducing a change in the repressor molecule that actually converts that repressor molecule into its inactive form. Remember, I said we were working with allosterically regulated proteins here. So, right, as we can see on the repressor molecule, we have an allosteric site for the inducer, and then when the inducer binds to the repressor molecule, it essentially converts it to its inactive form, essentially turns the enzyme off, very much like a non-competitive inhibition kind of situation. And then all you have to do if you want to turn the repressor back on, right, is remove the inducer molecule. So this is a totally reversible process. But if you remove the repressor, then remove the inducer, the repressor goes back to its active form, and now it can bind to the operator again, and we can turn that inducible operon back off. So inducible operons are usually coding for enzymes that are involved in catabolic pathways, right? And way back when, right, remember catabolic pathways, these are pathways where you're breaking something down, right? And in this case, this particular example is the lac operon. So we're coding for all of the enzymes, which you guys don't need to know this for lecture, but you may need to know it for lab. I'm not 100% sure. All of the enzymes involved in breaking down lactose, right? And you don't want to make the enzymes to break down lactose unless you actually have lactose, right? So this kind of explains why our inducible operons are usually turned off, right? So that regulatory gene that makes the repressor molecule is going to make it in its active form. And so basically the operator is always bound to the repressor. The gene is always, the operon is always off. except when lactose is present. And guess what? Guess who's the inducer? Allolactose. This is a form of lactose. So if you don't have lactose, you don't have allolactose. And if you don't have either one of those things, then you have an active form of the repressor. It's going to bind to the operator, and that whole operon is turned off. Again, it doesn't make sense to make enzymes that you don't need, right? You don't need to make beta-galactosidase if you don't have lactose to break down. So again, inducible operons are usually off, meaning the repressor is produced in its active form, and it takes an extra molecule, an inducer, to induce a change in the repressor molecule that converts it to its inactive form. And when our repressor is inactive, it can't bind to the operator, turns the whole shooting shebang on, and now transcription can happen. So if we have lactose, we want to be able to make the enzymes that let us break it down and get energy out of it. And so here we have, right, our operon is on, and so I'm going to, if I am RNA polymerase, right now I can start transcribing. I'm going to transcribe some extra junk code, and then here's my start codon. And interestingly, right, I've got 3 different genes. They're all going to be transcribed together as one piece of messenger RNA. And there's a start codon for the LAC-Z gene. There's the stop codon for the LAC-Z gene. There's the start codon for the LAC-Y gene. And again, the ribosomes in bacteria are very efficient, and they're basically just going to be translating this entire thing, and they're actually going to produce 3 polypeptides from this whole thing instead of 1. Okay, so basically the inverse of an inducible operon is a repressible operon, right? It's an operon that is able to be repressed, right? So you can think about that, right? Repressors are usually molecules that turn things off. And again, the active form of the repressor, even here in bacteria, it's the active form of the repressor turns the operon off. So again, this is very much like repressors act in eukaryotes too, right? They're binding to a DNA sequence and blocking transcription. So a repressible operon, I'm an operon that's able to be repressed. Well, if I can be repressed, it means that I'm usually turned on, right? I'm usually not repressed. I'm usually in the on position, right? And so, right, here's my regulatory protein and it with its individual promoter, right? It's always going to be on at a low level. It's always going to be making that repressor molecule at a low level, but you'll notice, right, here in this case, the protein being produced, right, the repressor molecule is made in the inactive form, right? And it's always the active form that binds to the operator and turns everything off, right? Remember, repressor's like an off switch. So my repressor has to be active to bind to the operator. If my repressor is produced in the inactive form, right, then my operon is usually turned on, right? And so transcription is normally happening. So here we have an example of a repressible operon, is the tryptophan or TRIP operon. And remembering, of course, tryptophan is one of our amino acids. So our repressible operons tend to be associated with anabolic pathways. You have to make something. And you usually need to make this something, whatever this something is. In this case, it's an amino acid that you need to make in order to make protein. And so usually the enzymes involved in this pathway are turned on, right? Usually our operon is on, meaning our operator is not bound to anything, and transcription can happen. And so typically transcription is happening, right? You're making all the enzymes, in this case there's 5, that are involved in the process of making tryptophan, that amino acid, from its component pieces. So this is an anabolic process, and normally these types of operons are turned on. So therefore, the repressor is produced in the inactive form. And so these are, again, normally anabolic pathways, and they're usually turned on. And it's going to take something extra, what we call a co-repressor, right? This little guy here, and to bind to the repressor and convert it to its active form. And again, in its active form, it's going to bind to the operator and stop transcription. It's always the active form that blocks transcription. What would be a good candidate to block the transcription of the enzymes involved in making tryptophan? Hopefully everybody answered to that question. They answered tryptophan, right? If you have it, you don't need to make it. Again, the whole point of gene regulation in both eukaryotes and prokaryotes, a large part of the point is don't make protein you don't need to make, right? Because making protein is expensive. So don't waste your cash and don't waste your energy doing that if you don't need to do it. If you already have tryptophan because you've stumbled across a food source that has tryptophan, then you need to turn off the enzymes that are making tryptophan because you don't need to make it because you already have it, right? Again, these tend to be anabolic pathways, so you don't need to build something if you already have that something. So in this case, that something or tryptophan is acting as our co-repressor. It's binding to the repressor molecule and converting it to its active form, and it's the active form again that's going to be bound to the operator and blocking transcription. Okay, so far we have a situation that's somewhat similar to eukaryotes, right, where we have a active repressor that binds to a DNA sequence to block transcription, basically turn genes off. And that's what we see in eukaryotes, right? We have repressor molecules that bind to, instead of binding to operators, they bind to those control elements, right? And so we have something slightly different in eukaryotes. And those control elements might actually also bind to activators depending on the kind of gene that's involved. And so you can use the same set of activators and repressors to turn certain genes on and turn other genes off. So we have a lot of flexibility in eukaryotes. In prokaryotes, right, we have the repressor molecule that's actually produced by a different gene. So it's not just any molecule that can serve as a repressor. It's only a specific type of molecule that's produced by a different gene. But it's still the active form that binds to the operator in this case and blocks transcription, right? And that's true for both inducible operons and repressible operons. It's always the active repressor molecule that binds to the operator and stops transcription, right? So an inducible operon, the repressor molecule is produced in the active form, and so it, am I getting this right? Yes. And so it binds to the operator, right, and turns the operon off. An inducible, you have to induce the change, right, in the repressor molecule, in this case, turning it off with an inducer molecule. And in the case of the lac operon, like we're showing here, the inducer is lactose, right? Because usually inducible operons are involved in catabolic pathways, and so it doesn't make sense to make their protein products, which are enzymes that are going to break something down, unless you have the something to break down, right? So it makes sense that their default position is basically off. Whereas a repressible, right, is the other way around, right? It's able to be repressed, so it's normally on, and that means that in that case, the repressor molecule is produced in the inactive form, and it requires a co-repressor to turn it on. And then, of course, it's always the active form that binds to the operator and blocks transcription. So again, in the tryptophan or the trip operon, it makes sense to always be on, meaning my repressor is produced in the inactive form, because I'm usually going to need to make tryptophan. It's an amino acid. I usually need it. But however, when I don't need it, I have it already, then I can use that as a co-repressor. It turns right, the repressor molecule into the active form, and it's the active form that binds to the operator and turns off the operon. Hopefully that's clear as mud. Okay, so what do we want to do if we're a bacteria and we want to turn up transcription, right? We want to actually transcribe a gene more than once, or an operon more than once. What do we want to do? What do we do? And so a very, very similar way to eukaryotes, right? We have molecules that are called activators. And the activator binds, in this case, it binds to the promoter directly. But in the case of eukaryotes, right, activators bind to those control elements. But in binding to the control elements, they increase the affinity of RNA polymerase for that particular promoter, and it transcribes it over and over and over again. Activators in bacteria, basically with operons, work in a really similar way. They are stimulating transcription, basically attracting RNA polymerase, increasing the affinity of RNA polymerase for a particular promoter, kind of acting like a magnet for RNA polymerase to that promoter by binding with the promoter itself. So rather than binding with control elements, these activators bind directly to the promoter, and in doing so, instead of blocking transcription, they actually increase it, right? So here we have an example of a particular type of activator, which is known as CAP, catabolite activator protein. And it is basically produced in the inactive form, and it's turned on, so we have another allosterically regulated protein. It's turned on by binding with cyclic AMP. Cyclic AMP is basically like your blinking low gas indicator, your fuel light indicator, right, that comes on and says, hey, you don't just need to get gas, you need to get gas like right now because you're in danger of running out of fuel. So this cyclic AMP is produced when, or is left over, right? You started with ATP, I hydrolyze a phosphate, I have ADP, right? I still have two more phosphates. I can hydrolyze ADP to make AMP, but now I only have one phosphate left. So cyclic AMP is basically a form of AMP, and you don't really have any phosphates left to hydrolyze. So basically, hey, I'm running out of energy. I don't have ATP, and I don't even have ADP. I'm running on vapor, right? I'm running on fumes. I'm running on AMP. And so when fuel is low, right, you're going to maybe turn up transcription. Hey, I've got a fuel, I'm very, very low fuel, and I have access to lactose. Hey, energy source, awesome. Let me make, right, the enzymes involved in breaking down lactose. So let me turn on my lac operon, right? Again, lac operon is an inducible operon, so it's usually off. So our repressor molecule is usually in the active form. It's turned off by allolactose, right, which is our inducer. And so therefore that frees up the operator and we have transcription happening. But in situations where you're really low on fuel and you have a fuel source present, you want to turn up the transcription of the enzymes that are involved in being able to break that fuel source down, because you need energy like ASAP. So that's where that cyclic AMP basically activates in this case, the catabolite activator protein, which is the activator for the lac operon. Other operons might have similar activators, or they might have different activators depending on what exactly they're producing, what their enzymes make. But in this particular case, the activator is activated itself by cyclic AMP, binds to the promoter, and in doing so, that turns up transcription of the enzymes involved in breaking down lactose. So essentially, when you have an activated, right, CAP, basically that catabolic activator protein, Once that's activated, right, it has a high affinity for the promoter, so it binds to the promoter, and that increases the affinity of RNA polymerase for that promoter as well, which turns up transcription sort of normal, more than the normal rate, right? So if you think of the repressor is basically like an on-off switch, right? If it's active form, it's bound, which means that the operon is off. If it's in the inactive form, then the operon is on. Then an activator is like the volume control, right? So without the activator, you sort of have a normal low level of transcription happening. And with the activator, that ramps up transcription, and you have RNA polymerase will, just like activators in eukaryotes, will bind to the promoter and transcribe that same operon over and over and over again. So you get extra copies. So it's very much like, you know, activators in eukaryotes. It functions in very much the same way, really. What's a little bit different is for eukaryotes, right? You can have activator or repressor molecules can be different things, right? Sometimes they're carbohydrates, sometimes they're other things. And every gene has, right, that set of enhancer regions with control elements. And some control elements respond to certain molecules as an activator, and some respond to the same thing as a repressor. And so you can actually turn a set of genes on and off with the same molecules. Likewise, all the genes in the same pathway in eukaryotes are on different chromosomes. And so they're all controlled by, they each have their own set of control elements, but they will have control elements in common with each other, so that you can turn all the genes in a pathway on at the same time or off at the same time, as the case may be. In prokaryotes, right, we have a repressor molecule that's produced by a particular gene, and that repressor only binds to the operator for that operon. It's not all, you notice we have inducible operons and repressible operons. The same repressor molecule will not act as a repressor only to all operons. It's just the specific one that goes with the gene that made it, right? It's a set. So you can't just respond to or have that repressor function with any operon. It's specific to the operon that goes with the gene that made it. And likewise, activators in prokaryotes tend to be very specific to which operons they're binding to. So it's a little bit, they're very specific and very efficient. Eukaryotes, we tend to be less specific and less efficient, but more flexible in our transcription, right? If you want to just make beta-galactosidase, but you don't want to make any of the other enzymes in the lac operon, you're out of luck. You have to make all of them, right? You don't really have a choice. They're all going to be transcribed and translated at the same time. Okay, so as I was just saying, in bacteria, right, which genes are turned on or off is going to be the result of interactions between repressors and operators, and of course, then, you know, which promoters are available to be transcribed and which aren't, and which activators are turned on and which aren't, and that's going to influence which operons are active and being transcribed, and which are being transcribed at a really high rate, and which ones are turned off, right? And in bacteria, you're a single cell. So while it's complicated, it's simpler than a eukaryote, where you may have, and again, not all eukaryote organisms are multicellular by any means, but in eukaryotes, because we have linear chromosomes and because of the way our DNA is organized, we have a lot more flexibility in turning genes on or off. We have a lot of other mechanisms for turning genes on or off than just repressors to turn on or off, and activators to ramp up transcription, which is mostly what bacteria are dealing with, right? Most of their genes are organized into operons, and you turn the whole operon off or you turn the whole operon on depending on whether the operator is free or bound to the repressor. And if you want to turn it way up, right, you have a free operator and an activator that's bound to the promoter, and that increases transcription. In eukaryotes, right, what we see on our little diagram here, it gets to be quite complicated because, right, we can, and this is something we've talked about in class, right, we can regulate transcription at the chromatin level, right, basically the whole chromosome level, right, we can turn an entire chromosome off, as is the case in exon activation in female mammals, right, we can bind methyl groups by those chromatin modifying enzymes to condensed chromatin, and that basically turns all of the genes on that chromosome or all of the genes on that part of the chromosome off completely, right? We can add a methyl group to individual promoters that blocks transcription completely, and that's going to turn that DNA off, that particular gene off. You can't transcribe it, right? We can add acetyl groups to the chromatin and actually open up that part of the chromatin and make it easier to be transcribed, and that's going to mean that those genes are probably going to be transcribed more often than other parts of the genome, right? We do a bunch of modifications to our oops, sorry, having a temporary issue. There we go. We can modify our messenger RNA that's made from a particular gene using, right, those spliceosomes. And the spliceosomes are directed by master regulatory genes, which are expressed based on the type of cell that a particular cell is, right, whether it's a neuron or a muscle cell or something else. And depending on what kind of cell it is, right, you get a different regulatory gene that's produced. And that regulatory gene will then direct the spliceosomes to treat different sections of a gene as introns and exons, depending on the cell type. So you can get from the same gene sequence, you can actually get multiple different proteins depending on which sections you treat as introns or exons. So you don't actually have to change the code itself. You change how you splice the primary transcript, right, based on cell type. So we make some modifications to our RNA that prokaryotes don't do, right? Prokaryotes, what's written is exactly what's written in the code is exactly what's read and exactly what's transcribed into the messenger RNA and exactly what's made. So you make it exactly like it's written in prokaryotes. In eukaryotes, that's almost rarely the case. And so this makes prokaryotes really great if we're trying to figure out what a gene does. In eukaryotes, we can give the prokaryotes the gene sequence and say, okay, make this. I want to find out what protein this even looks like. Now, knowing that it's not going to be the exact protein that's made in eukaryotes, but it might give me an idea of what that protein's job is, which gives me an idea of what that gene is for, right? So in eukaryotes, we often have a situation, again, all cells have the same DNA, but you want to express different regions, different genes in one cell type than you do in others, right? Depending on what job that cell has, right? And so this whole process of turning some genes on in some cells and the same genes off in other cells is what we call differential gene expression, right? So you can get, for example, it doesn't make sense for cells other than bone marrow cells or other cells that make red blood cells to make hemoglobin, right? My brain cells don't need to make hemoglobin. My brain cells don't need to make, some other stuff too. So it makes sense that in those brain cells, right, sorry, my cat is he's getting the zoomies, so that was the jingling. Sorry about that. You can have certain cells have those genes that they don't need turned off. And what's interesting, of course, is that the repressor molecules that turn those genes off, maybe sometimes permanently. We actually add a methyl group and just say, we're done, right? Master regulatory genes are also going to direct those chromatin modifying enzymes to methylate promoters. So you could permanently turn some genes off based on what type of cell you are. But in some cases, we turn some genes off and some genes on with the same set of molecules. Basically, the same molecule can act as a repressor for some genes and act as an activator for other genes, just to be cool and different. Okay, so that's basically gene control in bacteria.